During World War I, trench warfare emerged, introducing the utilization of a variety of lethal weapons, including a toxic yellow cloud known as mustard gas. Exposure to this poisonous gas has significant physiological impacts on anyone who came into contact with it, causing respiratory distress, eye burns, and the creation of large blisters upon skin contact. In an attempt to counteract the devastating impact of mustard gas, scientific endeavors were directed towards finding an antidote to mitigate its harmful effects. Interestingly, research into the toxicological features of mustard gas revealed that it has irreversible negative effects on bone marrow, specifically its hematopoietic function in the production of blood cells. This discovery encouraged researchers to investigate the medicinal possibilities of this knowledge. Since cancer cells and bone marrow cells share a characteristic of rapid replication, a novel approach to cancer treatment was envisioned. In the 1930s, research efforts were launched to investigate the viability of using mustard gas compounds for cancer treatment. This involves injecting chemicals obtained from mustard gas into cancer patients' veins. This investigation required careful trial and progressive development of methodologies in order to establish a balance between therapeutic efficacy and potential harm. By the end of World War II, researchers had identified a class of chemicals that would eventually be recognized as the first chemotherapy drugs. Currently, there are over 100 different formulations of chemotherapy medications available, most of which are injected intravenously or taken orally. These medications, which are referred to as cytotoxic agents, are toxic to living cells. Their primary mode of action damages all cellular entities within the body, even healthy ones, to various extents. However, their pronounced impact is observed in rapidly dividing cells, a hallmark characteristic of cancerous cells. A classic example of this idea can be seen in alkylating agents, the initial chemotherapy medications that are still used in cancer treatment today. When administered intravenously, these drugs spread throughout the circulatory system, gaining access to cells all throughout the body. Upon internalization within a cell, they induce damage to the fundamental building blocks of DNA's double helix configuration. The resulting disturbance in DNA replication endangers cellular viability, eventually leading to cell death unless appropriate repair mechanisms intervene. This strategy is justified by the fact that cancer cells are more vulnerable to the effects of alkylating chemicals. Cancer cells have an increased intake of these chemicals due to their rapid multiplication, making their DNA consistently vulnerable and inadequately repaired. As a result, cancer cells are more vulnerable to these assaults than non-malignant cells. The end result is a selective eradication of malignant cells with little influence on healthy cells with DNA repair capabilities and lower alkylating agent concentrations. Another noteworthy aspect of chemotherapy is the use of microtubule stabilizers. These chemicals exert their influence within cells by blocking microtubule disintegration, which is required for cellular division and DNA replication. Microtubule stabilizers obstruct the completion of cell replication through this method, ultimately leading to the death of cells. Microtubule stabilizers are representative of the 6 distinct groups of chemotherapeutic medicines used in cancer treatment today. Despite its tremendous therapeutic benefits, chemotherapy has limits. Notably, this technique has a negative impact on healthy cells, which have a high rate of renewal. Tissues such as hair follicles, the gastrointestinal lining, oral mucosa, reproductive organs, and bone marrow are subjected to significant stress similar to cancer cells because to their increased resource requirements and frequency of renewal. As a result, these tissues are equally vulnerable to the effects of chemotherapy medicines. This sensitivity supports a number of well-documented chemotherapy side effects, including hair loss, tiredness, infertility, nausea, and vomiting. In order to relieve the discomfort of nausea, doctors frequently prescribe strong antiemetic drugs. Furthermore, new therapies, such as cold caps, have been invented to reduce hair loss. Cold caps function by reducing local temperatures around the scalp, constricting blood vessels, and thereby limiting the exposure of hair follicles to chemotherapy agents. Upon the completion of a chemotherapy cycle, the injured healthy tissues go through a healing and restoration process, gradually resuming their normal renewal patterns. The long-term impact of chemotherapy is further demonstrated by the large number of people worldwide who are diagnosed with cancer each year, highlighting the seriousness of the illness. However, advances in treatment approaches, especially chemotherapy, have significantly improved the prognosis for many cancer patients. A remarkable example of chemotherapy's revolutionary potential can be found in the context of testicular cancer, where survival rates have increased to around 95% because of therapeutic advances. Similarly, in patients under the age of 60 with acute myeloid leukemia, a severe blood cancer, chemotherapy has produced remission rates reaching 60% during the initial treatment phase. Ongoing research efforts are devoted to the development of targeted therapies that only affect malignant cells, hence increasing survival rates while causing minimal harm to healthy tissues. This constant pursuit indicates the dedication to improving the efficacy of one of the most important tools in the overall fight against cancer. Thanks for watching. Hit the subscribe button to support our work.